Good afternoon. I'm Kanta Subbarao from the Laboratory of Infectious Diseases, NIEID, and I'm delighted to introduce today's Wednesday afternoon lecture series speaker, Dr. Peter Palese. Peter is a professor and chair of the Department of Microbiology at the Icahn Mount Sinai School of Medicine in New York. And Peter, we appreciate your taking the time to come here on a rainy day. Peter earned his master's and PhD at Vienna University in Austria and received postdoctoral training at the Roche Institute in New Jersey. He joined the faculty in the Department of Microbiology at Mount Sinai School of Medicine in 1971, became a professor in 1978, and chair in 1987. On a more personal note, I met Peter in 1990 when I joined Brian Murphy's lab as a postdoc, and we collaborated on a project to put influenza B ends on an influenza A gene. Peter has since gone out of his way to be helpful to me at different stages of my career, so thank you. Peter has received numerous awards and honors during his career, among them the Howard Taylor Ricketts Award from the University of Chicago in 2005, the Robert Koch Prize in Berlin in 2006, the European Virology Award from the European Society for Virology in 2010, and the Sanofi Institute Pasteur Award in 2012. In 1998, he was elected Fellow of the American Association for the Advancement of Science, and in 2000, Fellow of the American Academy of Microbiology and member of the National Academies of Sciences. In 2012, he was elected to the Institute of Medicine of the National Academies of Sciences. In 2005, he served as the president of the American Society for Virology. Dr. Palese's research has primarily been in the area of influenza viruses. Specifically, he established the first genetic maps of influenza A, B, and C viruses, identified the function of several influenza virus genes, and defined the mechanism of neuraminidase inhibitors. He was a pioneer in the field of reverse genetics for negative strand RNA viruses, which allows the introduction of site-specific mutations into the genomes of these viruses. This technique has revolutionized studies of the structure and function, relationships of viral genes, investigation of viral pathogenicity, and the development of vaccines. Most recently, he has turned his attention to the topic of today's lecture, which is towards a universal influenza virus vaccine. Please join me in welcoming our speaker for today's lecture. Peter.
Thank you very much, Kanta, for the very, very nice introduction, and I'm very happy to see so many friends here. The last time I was actually in this room was when George Bush gave a lecture, and he got a standing ovation at the end. So I hope you will be equally tolerant to me. Can we make an influenza virus vaccine? So I will be very specific about this one topic, and we'll have a short introduction. So this is the virus. It's an RNA-containing virus. There is an RNA in the center, and there are 7 RNAs around it. And then we have a lipid membrane. And what is really recognized by the immune system are the surface glycoproteins, the hemagglutinin, and the neuraminidase, and they come in different subtypes, and we will talk about this a little bit more as we go into the subject. What is also important to know is that these RNA segments are sort of like mini chromosomes, so if we have 2 of these viruses infect the same cells, one can get an exchange of these RNA segments, and viruses come out which have maybe 7 of one and 1 of the other, and you can see that there is a lot of genetic exchange possible, and that is giving rise to what we refer to as pandemic influenza viruses, and we will also discuss this in more detail. Now, what is the subject of today's talk is that we believe we can make better vaccines. So the present vaccines against influenza are pretty good, but they have to be given every year, and as we will discuss, there are some other problems associated with them. So we want to make a vaccine which can last for 20 years or even a lifetime. And the principle of this is, so the hemagglutinin, one of these, or the major glycoprotein on the surface of the virus, this hemagglutinin has a very much conserved domain, but a head which is much more variable. And actually, the head is 10 times more variable than the stalk. So we want to make a vaccine, basically, where the immune system is directed against the stalk of the hemagglutinin as well against the neuraminidase, which is the second, much more conserved protein in the virus. So that's the principle we want to make. When the regular influenza virus vaccine is given, most of the antibodies are made against the head, which is more immunodominant. We want to make an immune response against the subdominant stalk of the hemagglutinin and the immunosubdominant neuraminidase. Okay, just a little bit more background. We have many different subtypes of the hemagglutinin. There's actually now a total of 18 different subtypes, and they are very, very different. If you look here at the scale, this is 4% down here, and so you can, if you add up these distances, we can really clearly see that some of these hemagglutinins have only a sequence conservation of about 40–50%. So these 18 different hemagglutinin fall into two major groups, a group 1 on the top and a group 2 on the bottom. But what is very important is that since 1918, we only had H1, H2, and H3. So these are the ones which are boxed in red. And so the H1 and H2 belong to the group 1 of the hemagglutinin, and the H3 belongs to group 2, and that will become important as we go on. And then just sort of to give you a feeling about the neuraminidase, we have 9 different subtypes at this particular, or 10 different subtypes at this point, and only 2 have appeared over the last 100 years, N1 and N2. Nevertheless, the big problem with influenza viruses is that they are changing. So there is a lot of variability. If we talk about a measles virus vaccine, mumps virus vaccine, we are using the same vaccine which was developed in the 50s. So the virus has now changed. Otherwise, the vaccine wouldn't work today. With influenza, this is different. We have a continuing change from year to year. We have antigenic drift, or even we can have an antigenic shift, which is when a new RNA, after this reassortment, which I briefly described, appears, and then we have these pandemic strains, which are associated with much more disease and much more morbidity and mortality. And so this is sort of a rather complicated slide which shows the different influenza viruses over the last more or less 100 years. And we have influenza A viruses and influenza B. There's also a third type, influenza C, but that is usually not causing a lot of disease. And what we can see here on the left is the 1918, and that was based on Jeffrey Taubenberger's sequence, which he obtained by very elegantly sequencing material from patients who died at that time. And in collaboration, we were able to actually reconstruct in the laboratory such a virus. So the virus is extinct, but the H1N1 from 1918 has given us a lot of information why this pandemic virus was so bad. And this is sort of a hallmark. More than 50, probably up to 100 million people died in this 1918-1919 pandemic. And then what we see is that the virus was present in H1N1. Remember, H1 belongs to the group 1 of the hemagglutinins, and the virus changes continuously. That's why I have sort of changed the color here in terms of from starting 1918. We have the first viruses, human viruses are going back to 1933. And then in 1957, we suddenly got an H2, so that's a new pandemic strain. And that one was with us only 11 years. And again, you see this color change, which means that after 2 years, 3 years, the virus has sufficiently changed, antigenic drift, so that we can get disease again, and we can actually really reinfect it first and then really getting disease. And this is then accentuated if we get a new pandemic strain, as in 1968. This is an H3N2 virus, and this actually, this subtype of H3N2 is still with us in 2014. And very strangely, we had a reappearance of in 1977 of basically 1950 strain. And again, this H1N1 was with us until 2009, changing, and that's sort of shown in this color change. And then we had something very, very strange happening, that we had a pandemic virus of the same H1N1, a subtype as we had this what we refer to as a seasonal H1N1, but the head of this pandemic H1 was very different, and you will see why this is of importance. And we believe that one of the very good explanations why the pandemic H1N1 really led to a rapid extinction of the old H1N1 was because some stalk-specific antibodies were introduced. And I will explain this in more detail, but we believe that the extinction of the regular H1N1 in 2009 had to do with a very interesting induction of antibodies which were recognizing the stalk of the old seasonal H1N1. Now, I mentioned the pandemic of 1918, and that always is sort of in the back of people's mind when we talk about influenza. I think this is a very different story. Today, one is that we have antibiotics, so the superinfections by bacteria wouldn't be so bad. But also what we have learned, and these are studies in Taubenberger's lab, in Terry Tumpy's lab, ours lab, Adolfo Garcia Soster's lab, we find that the virus is actually sensitive to drugs, to neuraminidase inhibitors, but also even the older amantadine derivatives. And more importantly, we all have some antibodies now against these H1 viruses, and so that even if such a virus would be found in humans, it would not have the same impact as the 1918 H1N1. So it's not only that, and this virus was very virulent, that is by all means and all the experiments we can do in the laboratory, this was a real virulent virus, but it always depends also on the immune experience in the host, and clearly in the 1918 must have been a very different situation. So I think by learning about what this 1918 virus was, we, I think, are in a much better position now to be prepared if something else of that ilk would happen. Okay, now, in addition to these H1, H2, H3, so those are the only three subtypes which we have seen and have experienced as humans in the last 1919 period on, there are many avian influenza viruses which have been identified, and then just last year in China, all in China, and that has probably to do because half the population of the world lives in this area. We had H10N8, H7N9, and H6N1 all in 2013. And if you look a little bit more carefully, you can see actually that there were other H10 viruses before, like this H10N7 in Australia and Egypt, 2010, another H7N3 and H7N2 before that, and down here in H7N7. So we have occasionally, and this, I think, has to do with the increased surveillance which we have had over the last 1 to 2 decades. We see that some of these viruses actually can jump from avian populations, mostly chicken, into humans. And particularly the H5N1 has caused a lot of problems, and starting in first appearing in 1997 and then since 2003. However, none of these different avian viruses with these different hemagglutinins against which we humans do not have any kind of immunity and protection, nevertheless, these viruses do not have the capability so far to really jump from one human to the other. And in most instances, it is likely that only a large dose, 10 to the 6, 10 to the 7 infectious particles, is a bolus, has been inhaled by people who were animal handlers in live chicken markets or had to do with chicken in rural areas. So in this case, I think all of these viruses, even though they have been found in humans, I think there is not the greatest likelihood that these will be the next pandemic strains. Now, we have sort of asked the question, why do these viruses change so dramatically in terms of why is this antigenic drift so observable in humans and not, for example, with other viruses? And we did, and this is work by a postdoctoral fellow, Nick Heaton, who did an interesting analysis, and this is just one slide, to show us why we believe that the hemagglutinin is such a unique molecule. And what he did was, on the left side, we see a monomer of the hemagglutinin, and one can sort of divide this into a globular and into a stalk domain. And what he did is he mutagenized by transposon mutagenesis, inserted into every position of the about 1,800 nucleotides, a 50-nucleotide long piece. And then we asked the question, can we rescue viruses by reverse genetics which are stable? And then it turns out, very, very interesting, that, and I give you the bottom line, basically all the mutations were just in the head. And this can be shown here. So this is the globular head, this is the stalk here, and the stalk is green here, so they're the first 52 amino acids, then going on from 277 to 565. And so we are looking here at the input, so basically we can't show all the colors here, but in essence, we were able to show that mutagenesis worked, that in essence almost every nucleotide position was changed by introduction of 15 nucleotides. And then after the rescue, we put this into eggs, rescue in eggs. And now we look here, what was the majority or what came out, what was rescuable and what was rescued. And if you look here carefully, so we have this blue here, and the sequence falls right into here, into the globular head. And then we have a light blue here, not that much, another blue, this blue here, and yellow, red, and this yellow here. And all of this, all the way down here, if you look, go back here, these are all in the globular head. All of these, so these are all 15 nucleotide insertions, 5 amino acids. So this is a very robust mutagenesis and tells us that the head of the globular head is much more tolerant, much more flexible in terms of mutagenesis. And that the stalk really doesn't allow us to make these mutations. So this is sort of an explanation why we believe that the stalk is much more conserved as compared to the head. And it has to do that the stalk is involved in fusion, and it has to sort of more functions. It's more important that there are no changes. So this is really a very recent experiment. This just came out in PNAS and allowed us to say why we believe that there is so much genetic variation in the globular head. And even if you look down here, there's another blue one. But if you look all the way down here, this is an insertion outside of the coding sequence. So this is really not reflected in the protein. So this is, one can get an introduction of 50 nucleotides past the stop codon. So this is really, I think, a quite interesting result, and we are doing experiments right now with measles virus, because we believe in measles virus we cannot select such transposon mutants because they won't tolerate. Otherwise, I would feel we should be able, or the virus would be able to tolerate changes. So just sort of, we suggest that the head domain of the hemagglutinin is uniquely tolerant to mutations, and this is sort of an explanation for the rapid antigenic drift, and also at the same time explains why we don't see this with other viruses, but those data we don't have yet. Okay, so let's talk specifically about the vaccines which we have. Again, we have pretty good vaccines available right now, and they fall into two categories. Killed, meaning they are usually formaldehyde treated virus, and then it is split to make it less reactogenic. And we have also live attenuated vaccines, which is a cold adapted vaccine, and that is usually given through the nose. So we have these two flavors of vaccines: killed, administered by needle IM, or we have by syringe a live virus vaccine, and that's a cold adapted. So that's what we have right now. And as I mentioned before, we have vaccines which have to be changed actually every year, and this is not that complicated, just look at the colors here. So we have here the vaccine formulations of the vaccines which were used from 2000 to 2010, and red means the vaccine, this particular component, so we have an H1 component, an H3 component, and a B component, and red means the strain was changed from the previous year. So the first one here, 2 components were changed. In this case here, all 3 had to be changed in 2008, 2009, and only 1 year, 2003 to 2004, is identical to the previous year from 2002 to 2003. So this is complicated. We sometimes don't hit the right vaccine strain, and it is quite cumbersome to prepare this every year. And also in a case of a pandemic, and these are CDC data, where one can see that we were too late in 2009 when this pandemic occurred. And the red curve shows when we had the maximum influenza activity in the country, and the blue one was when the vaccine was shipped out. So it wasn't even distributed yet, and then both live and killed, it takes about 10 days until they're really effective. So you can see that we were much too late. Fortunately, this pandemic 2009 H1N1 virus was not very highly virulent. Otherwise, we would have had a real problem, because the vaccine, I mean, basically we would have to shift it until it worked by another probably 4 or 5 weeks, and clearly we would have been too late. So that's a problem with our current vaccine. It takes too long, and I can easily discuss this in more detail, why it takes so long, but we have to really look at the strains which are circulating in the southern hemisphere in July and August, and then we make up our mind. Well, the CDC makes recommendations, the FDA accepts or defines then what the new strain should be, and it has to be changed, and particularly with respect to a killed one, it is not good. With respect to a pandemic one, we were too late. And also, this was particularly last year, where we had mostly an H3 year, and in that case, the H3 vaccine strain didn't really match what was going around, and the hospitalizations, more than 20% of all the hospitalizations in the last 2 weeks of January were associated with influenza infections. So there are really questions in terms of, can we improve on it? And the question is, how can we do better? And that's the sort of concept of a universal influenza virus vaccine. The idea is not a new one. People have tried basically in the past to look for domains and epitopes which are conserved. And so here we have several of the universal influenza virus vaccine approaches in the past. One was the M2, which is a very small protein, but it's very much conserved. And we have also looked at the neuraminidase, or people have looked at the neuraminidase, and I think that's a very important component of a universal influenza virus vaccines. There have been attempts to actually build universal virus vaccines on CTL epitopes, and also there's passive administration of antibodies, but that obviously would not be feasible in terms of a vaccine for everyone. It's sort of possible for people in intensive care units, etc. But what I want to sort of concentrate on is to the concept of redirecting the immune response to the stalk region of the hemagglutinin and also the highly conserved neuraminidase. Now, in terms of hemagglutinin stalk-specific antibodies, in the last several years, we have actually identified mostly Lanzavecchia, Ian Wilson, the group around Crucell, have identified human monoclonal antibodies, Wayne Marusco at Harvard, which are binding not to the head of the hemagglutinin, which varies a lot, but rather to the stalk. And then we have also identified or designed a vaccine approach in mice where we have vaccinated with we did it with an H3 first, where we had one stalk and one head, and then we vaccinated that same mouse with basically the same stalk and a different head, and the third time with the same stalk and a different head. And what we got out was mostly antibodies against the conserved stalk. So that was an experiment in mice, and such specificities against the stalk can be easily seen if one does a mouse experiment and vaccinating in the way I described it. Now, the most important, however, is what does nature do? And we believe that nature uses this mechanism of directing antibodies against the stalk, and that this was the mechanism which explains the elimination of the earlier strain in terms of the H1N1, the seasonal H1N1, by the pandemic H1 in 2009. And so very briefly, what we are proposing is that when we have a seasonal vaccine, this is pre-2009, and we are having a stalk here and a head, and the head is always more immunodominant, probably or possibly because it's sort of really on the outside. That's what the immune system sees first. And then when one gets infected or vaccinated, one makes mostly antibodies against the stalk, against the head, but not against the stalk. However, what happened in 2009 was that we had again exposure to the pre-2009 seasonal H1N1, and then the pandemic H1N1 came about, which had, as I said before, 20% difference in the head and more or less conserved stalk. And what we believe happened was that this was sort of, we were naive against this new head, but we had then a recall and an amplification of the B memory cells against the stalk, and we found that actually the average of people who were infected with this new pandemic H1 showed an increased stalk titer of about 15 times. And we believe that this contributed to the elimination of the seasonal H1N1 by the popping up of this pandemic one. And this is mostly by Nelly Pica, a very gifted MD PhD student. So how are we going to make this vaccine? And we actually had to develop assays to measure these stalk antibodies, but also to make antigens. And what we found, and just remember, we look again here at a monomer of the hemagglutinin. We have here a head, we have here a stalk domain. And so the nomenclature is a little bit different. Here now we have a chimeric H6, so we have a completely different subtype. Remember the second slide where I showed the different hemagglutinin subtypes. So we make now a chimeric structure of the hemagglutinin where the head is at H6, but the stalk is actually from an H1. And this one here would be an H9, and this would be a stalk again from the H1. So with a chimeric H9/1 means it has a chimeric, it has a head from H9 subtype influenza hemagglutinin and a stalk from subtype 1. And in terms of the linear sequences, again, we have this cysteine in position 52 and 277, where we exchange the H6, and we can not only exchange it into an H6 or an H9. We have made now dozens of different ones. So for example, these are now, we are using different stalks. So this would be a PR8 stalk. This would be the Cal9, which is in the present 2014 vaccine. And then we have here the head, H1, H2, etc. And then we have here different stalks, and also we have different neuraminidase. So we have a lot of these different viruses, and not only in terms of the head from group 1, but also group 2. So we have here a head from group 2, for example, H7, with a stalk from group 1, either PR8, which is 1934, or the 2014 vaccine component. And then we have different neuraminases here. And sort of you can see the sort of direction we are going. We have here now, for example, an H5 with a different, H5 is also group 1 with a group 2 stalk, and here we have group 2 heads with group 2 stalks. So what is remarkable is that these viruses grow very well. So we have, for example, here, so those who have dealt with hemagglutination inhibition titers. So this red blood cell on the bottom is negative, and these are positive. So you can see we have a virus now which has, again, an H5 head, a PR8 stalk. In this case, it's actually a Cal9 stalk, and then this grows really out to 10 wells. So this is pretty good. This is like 10 to the 8, 10 to the 5 times 10 to the 8 particles. And those who have plucked viruses will love that, because this is PR8, which is sort of the house animal which everyone is using in the laboratory. It grows very well in eggs, and these are the plugs which are formed. And so this chimeric H5N1 Cal9, for example, is almost indistinguishable in terms of its ability to grow one, but also to form plugs. Okay, so what have I said so far? A brief summary. We have developed analytical tools, namely these viruses, if they have an irrelevant head, can be used to, and an irrelevant neuraminidase, if we check whether they can be inhibited by neutralization assays or by ELISAs, they will only bind if we have antibodies against the stalk. And also, we can use them not only to detect and quantify, but also we can have reagents which can be used to neutralize influenza viruses from different heads and containing different heads. And I will show several data in terms of unfortunately not humans, in terms of animals. I will show data in terms of mice and in terms of ferrets. Okay, and then also I think that's a very important aspect, is that we humans do make these stalk antibodies, and the increased titer of stalk antibodies in the population infected with the pandemic H1N1 serve as an explanation why the previous H1N1 viruses have disappeared from the circulation. And so now the next couple of slides will be in terms of what have we done to show that this can work. So what the principle is, we have all pre-existing immunity. For example, in H1, we have not as much stalk antibodies, but we have all head antibodies. And then the principle is we would come with a vaccine which is identical to the present vaccine, except that it has an irrelevant different head. So it would have the same 2014 H1, and we would boost with this chimeric H5/1 hemagglutinin, so that we would amplify the B cells, the B memory cells against the stalk, and then we boost the second time with a chimeric, in this case H6, but it could be H7, it could be H8, H9, because we don't have any immunity against any of these different heads. And in each case here, we hope that we get enough of this anti-stalk antibody enhanced in terms of a protective immune response. So if we go now to data which we have in the mouse, we have to, mice are completely naive, they've never been infected, so what we are doing is, and we don't have to do this in humans, we are priming them with a chimeric H9/1 DNA. So in this case, we have no neuraminidase because we want to study the effect of the stalk in the mouse. And then we boost with a CH6-1 and a CH5-1. So the only common denominator is here the stalk in each case. And then we challenge with different viruses. So we prime here, we get some antibodies against the head, some antibodies against the stalk. Then we boost, we should get, again, some antibodies against the head, but more antibodies against the stalk. And the second boost actually will give yet a different head, and we get here even more stalk antibodies. And then we are challenging, and this is now very important. So we have here, so the stalk here was against the 1934 virus. Then we use its challenge in 1947, or a challenge in 2009, or we use a completely different subtype, H5N1 and H6. So all of these animals have just seen protein. They have not seen anything else. And what we can very nicely see is that the vaccination with these chimeric HA constructs allow a complete protection. In this case, for example, I just showed the 2009. So we used the 1934 stalk, and we have here mice. We have starting with an initial weight. If we have no vaccination after 7 to 10 days, all the mice are dead. In terms of the regular vaccine, obviously, if we are working in terms of a homologous Netherlands vaccination, that should work too. But more importantly is here a vaccine based on a much earlier 1934 stalk also works very well. And if we look here at the survival, all of the animals survive. You can clearly see this here. So again, what we have been able to show is that a stalk immunization with a stalk from 1934 protects against, in the mouse, against the challenge of a 2009 virus, and it is basically as good as the homologous vaccination. And more important is that we can also challenge now with a different subtype virus. And again, we use here an H5 or an H6, and these were the same animals having been immunized in the same way. We first the DNA priming, and then we have boosting here with these two different chimeric proteins, and the increase in titer is we can measure in terms of ELISA assay, and we also just show one slide here so that we have here we measure the stalk antibody, which is very good, and more importantly, we can also passive transfer of serum protects from viral challenge. So this is clearly telling us something about the mechanism that it is mediated by antibody. And we can also do the same thing with ferrets, which we have done with mice, and in that case, again, the red are the chimeric HA vaccinated animals, and you can see here there's quite a drop in, for example, olfactory bulb titers, nasal turbinates, lung titers, etc. And we can also demonstrate that the mediation or that the mechanism is the same as we saw in the mouse, because we can show that the vaccination of these ferrets induces a broadly reactive antibody, and we find the red curve is, for example, we have Cal-9 antibodies against the stalk. This one, I think, is a, okay, this would be the 1918. This would be, I think, South Carolina. No, it can't be. This would be an H2, and this would be an H17, which is actually only a bat virus, and no virus is isolated. So this is just based on the sequence. So we can clearly see that also the ferret makes these antibodies directed against the stalk of the hemagglutinin and provides protection. And then, obviously, if you remember, we have group 1 hemagglutinins, but we also have group 2 hemagglutinins, and we have these H3, so that's the only one we have so far, but H7, and that's sort of one of the viruses which have popped up in China, and that belongs to the same group. And so what we did is we did a vaccination now. We tested first whether there is any evidence that the group 1 vaccinated animals are protected against the group 2 virus, and that is not the case, which would be on the left side. So we have here animals which were vaccinated the same way, identical to what I said before, namely against the PR8, a group 1 stalk, then boosted with a chimeric H6, again with a group 1 stalk, and here with a group 1 stalk. And when we look at this in terms of antibodies against the group 2 stalk, we don't see anything. And more importantly, in a challenge experiment, again, the H3 virus is not, as a challenge virus, is unfortunately killing all of these animals. So that means for a truly universal vaccine, we have to have a group 1 stalk as well as a group 2 stalk. And we have shown this now. Again, I only show the data in terms of the mouse, where we have here, we prime here now with a group 2 stalk, so that would be from an H3 influenza virus, has a H4 head, and then become a group 2 stalk again, different head, group 2 stalk, a different head, and then we challenge with, there's a Philippine strain in H3, 1968 H3, but also H7 and 9, and I wouldn't show the data unless I can show you that we get protection now in mice which have been immunized with an H3 stalk, which is group 2 stalk, and we get complete protection also against the different subtype, in this case the H7, so that all animals are protected in terms of survival here. And so the last slide is just sort of a summary of what we have done in terms of understanding the mechanism. So the stalk-specific antibodies can inhibit hemagglutinin-mediated fusion, and there are some assays which can be used. We can also show that the cleavage, and this is not only from our data, but also Ian Wilson, and that the trypsin-mediated cleavage of the hemagglutinin, which is necessary for virus replication, can be inhibited. Very recently in a Nature Medicine paper, together with Jeff Ravitch from the Rockefeller University, we showed that these stalk-specific antibodies actually engage Fc receptors, particularly macrophages and NK cells, and that's one of the mechanisms by which, probably one of the most important mechanisms by which these stalk antibodies work. They also activate complement, and also actually very similar to influenza virus antibodies, they actually also inhibit viral egress. So we are spending a lot of time to really understand what these antibodies are doing and what their mechanism is. And the last slide is here. What are our conclusions? We have an influenza virus vaccine approach here, which we hope will reduce the immunodominance of the head of the hemagglutinin and enhance the immunogenicity of the HA stalk, and as well as that of the neuraminidase. And we want to achieve this through chimeric hemagglutinin constructs, which are not inducing as much of an antibody and sort of redirect the immune response towards the stalk as well as the neuraminidase. And we have shown in the animal, so these are all animal data only, that we can protect against the challenge with heterologous, meaning variants of the same subtype, but also going from one subtype to the other, as long as it is in group 1 with group 1 stalk vaccinated animals, and against viruses of group 2 with stalk 2 vaccinated animals. The observed protection is antibody-mediated, and we can also show good protection against potential pandemic viruses such as H5N1 and the H7 and 9. Let me close here, and I'm happy to take some questions. Or there are no questions? Okay.
So we have time for questions, and there are 2 microphones back there. And I would like to remind you that there's a reception in the library right after this.
Yes, thank you, Dr. Palese. That's really wonderful. Do we know, or can we say, or can we imagine a timeline to the phase 1 trial?
I was told early on I should never give any timelines.
Okay, okay, fair enough.
No, but it's an effort which is driven by BARDA and NIH, and I'm very hopeful that this will get into people pretty soon.
Okay, thank you.
But this is not yet a vaccine.
So if it is a universal vaccine, do we need vaccination every year? How long does it cover?
Okay. So clearly, the idea of a universal influenza virus vaccine would be that we don't have to vaccinate every year. Yeah? So we would hope that based on the change which we see in the stalk, we would love to suggest it would last 20 years or even for a lifetime. In the animal, we can have even a longer period. Yeah? Where, I pointed this out, 1934 versus 2009, so that would be an even longer one. Yeah? But the universal vaccine approach is that we only would need one, hopefully at least 20 years and maybe a whole lifetime. And that we need 2 doses is not for those who are physicians here. They are using it all the time. You know? I mean, to give 2 vaccine doses is not really the end of the world. You know?
So when the clinical trial is starting or will be starting?
I mean, it has to go first phase 1, phase 2, phase 3, yes. Yeah.
Very exciting. I was just wondering, for the cell biology of the neutralization, do you know whether it has to take place before binding and entry, or is it possible when the stalk is more exposed in the endosome after acidification, there might be traveling of antibodies into the endosome with the virus?
Yeah. So we want to induce a polyclonal antibody response, which would include, but mostly towards the stalk. So that would include antibodies which recognize it on the virus itself. And Dr. Subramanian has shown actually that such antibodies can bind to about 3/4 of the virus, to the hemagglutinins of three-quarters of the hemagglutinin spikes on a virus itself. So we would argue that some of them is on the outside already, but when it gets taken in, then it would also prevent fusion, for example. Yeah? And also some of these antibodies may be where it binds to the virus, and with the Fc receptor, it binds an NK cells, which then come and may eat it up, or a macrophage. So I think my answer would be all of the above, we hope.
Right, because we've always been bemused by the fact that even if we make a peptide antibody against a fragment of the hemagglutinin that should be buried, like the fusion peptide, it turns out to be not such a bad antibody for intact PH7 virus.
Yes, that we have some breathing going on, yeah, so that a millisecond it opens it up and then it can bind the antibody. So we would argue that this happens, you know, this should happen with those antibodies as well.
Very interesting. What is the antibody profile, and do the animal develop allergies? That's one question. The other question is, do you have any differences in the effectiveness of these viruses toward aging, if it is done in the younger animal or older animal?
Okay, so the first one, the animal system we have used with our vaccination, we have only done mouse, guinea pig, and ferrets. And there we have not seen any untowards effect whatsoever. And literally, we have now vaccinated thousands of mice. Yeah? And based on the fact also that we humans do make those antibodies, that we don't, it's not that we don't make any, and particularly in the case of the pandemic H1N1 in 2009, we actually make quite a lot. And nothing has happened really, in essence. So I feel that this is probably not so much of a problem. And the second question was?
The age.
And the age, no, we have not looked at that, okay? We are a poor academic laboratory, so it's...
Thank you.
I just have to needle the NIH a little bit. Yes?
Thanks for a very nice presentation. Understanding that the stalk is not immunodominant, nonetheless there's some naturally occurring anti-stem antibody. Why is that not protective against pandemic influenza?
Okay, I would argue that we are now making in the population actually some of the antibodies, stalk antibodies, which eliminate the old one, and I would predict, and I'm going out on a limb, that this H1N1 will not last long, because we will make more of these antibodies, and basically I would argue this H1N1 will not be around with us for much longer. Okay, so this would go along with the idea that, yes, we make those antibodies and they should be protective. And in a way, the H2, where I think the same thing happened going from H1 to H2, the H2 only lasted 11 years. So if you have a group 1 and a group 2, they can easily coexist. But we will never have really two group 1 or two group 2 viruses at the same time circulating. And I would argue the present H1 will die out soon.
Peter, we've had a number of clinical trials with H5, H7, and certain other subtypes of vaccines. Do people who receive those vaccines, have they made more stem antibody?
Yeah. So we're only starting now. We have some data from Norway where we can measure that, and we have also some data from from protein sciences, where there is a very good increase in antibodies. Whether it's enough, we don't know yet. But these are only one. I mean, we would hope by having 2 vaccinations with different heads that we would be even better. And the head is not right. The head is an H5 head and an H7. We want a 2014 stalk and a 2014 neuraminidase.
How concerned are you that you'll drive escape mutants in the stalk region with these vaccines?
Yeah, no, it's a very good question. Why wouldn't that stalk also mutate enough? And yes, the answer is such evasion mutants will happen. It's a polyclonal response, though, which is a little bit more difficult. But we would argue that, or we would hope that these stalk mutants would be less fit than the wild type, and that we would be able to last at least 20 years, hopefully longer. If that happens, then we would need a new vaccine with a new stalk. But we would hope that the stalk is not as flexible and tolerant as the head is, so that these mutations would be taxed then by a fitness problem and would not be able to transmit as easily, so that they would not survive as easily. And the hope is also we are really banking on neuraminidase antibodies as well as stalk antibodies, and together this is about 800 amino acids. And so we would hope that there would be enough antibodies against these different epitopes, so that even one escape mutant would happen, it wouldn't really change much. But it is clearly one of the concerns. Yeah.
Any more questions? If not, I invite you to join us in the library for a reception, and thank you, Peter, for